Hey guys, we're going to learn today about how to do some Punnett squares. And so we're going to look first at Mendelian genetics. Basically, genetics and heredity for a really long time was thought of as the same thing. So they basically both deal with inheritance and inheritance of traits that you may find. So as we've progressed, we basically have figured out how traits get passed down from parent to offspring. And sometimes you have a situation where you have more characteristics of your mom or more characteristics show up from your dad. And then sometimes there's a combination of the two. The first person who really worked with genetics and how traits was passed from one parent to the next was Gregor Mendel. He is known as the father of genetics. He was a monk and he was living in Germany. He was born in 1822 and he lived in the Czech Republic and he tended a garden of peas. And so he did a lot of different experiments with his peas. He worked with the peas for 7 years. He published his results in 1866, but people didn't really pick up on the information or really even put it into practice until around the 1900s. So first of all, what is a trait? A trait is a characteristic that can vary from individual to individual, and you can actually see the traits that he chose. There were 7 pea plant traits that he chose to study. He studied the seed color, the seed shape, the flower color, the pod shape, the pod color, the flower position, and the plant height was the things that he chose to study. He was lucky that each trait happened to be located on different chromosomes. He didn't know about chromosomes back then, so he couldn't have purposely done that, but he was lucky that it turned out that way. So the first cross that he made was considered to be a pure breeding or true cross, and then he experimented with different traits one at a time. He cross-pollinated plants, which means he took different traits and he observed the offspring from these traits being crossed. Normally, this is done by our insects, like our bees, but Mendel used scissors and a little brush to help him to transplant them. So some of the vocabulary that you're going to need to know for this unit is pure breeding, which is where the offspring always have the same trait as the parents every time, no matter what. Self-pollination, which means the plant fertilizes themselves. Cross-pollination, where you take one, like the pollen from one plant and you fertilize another plant. Your P generation, which is your parent generation. Your F1 generation, which is your first generation of offspring, basically mating and seeing what the results are from that cross. And your F2 generation is your second generation offspring, which you basically take and have the creation from your two of your F1 generations being crossed. So when Mendel crossed the true breeding tall plants with the true breeding dwarf plants, he basically thought, let's see what happens. And all the plants were tall. There were no short plants. So that was sort of interesting. There wasn't a combination of the two traits where you would get a medium plant. It was just all tall plants. So Mendel was surprised, and he had expected the traits to blend. So he expected a lot of medium traits. But instead, the tall parents had made the short parents disappear as far as traits were concerned. So they sort of masked them. So then Mendel let the F1 generation self-pollinate to create F2 generation. And some of the F2 offspring were tall and some were dwarf. And the ratio always appeared as a 3-to-1 ratio. 3 tall to 1 dwarf. So when Mendel crossed true breeding plants with different traits, all of the F1 plants showed only one trait, tall. When you let the F1s cross, they showed a 3-to-1 ratio. And he didn't get any medium plants on either of the crosses that he had done. So he realized that something was masking, one trait was masking another trait. So he called the trait that was doing the masking dominant, which is what showed up in the F1 generation. And he chose the one that was being masked as calling it recessive, because that one didn't show up in the F1 generation. It was sort of hidden. So this is what he basically got. He got the parent generation. Something happened. Hold on a second. The parent generation, he crossed purple and white with the plants. And then he got all the plants that were purple when he was looking at the flowers. And then he, for F2 generation, he crossed the two of the F1 generation. And he got 3 purple plants to 1 white plant, 3-to-1 ratio. So he basically realized, and he looked at the color, and he said yellow mating with green. First generation was all yellow. Second generation, 3 yellow to 1 green. So his conclusion is that he realized that inheritance of trait is controlled by genes contributed by each parent. And these genes came in different forms called alleles. So some of the alleles are dominant while others are recessive. And when both types of alleles are present, dominant alleles will always be the one that appears. So this came to him creating what's called the law of dominance. So generally, one allele is dominant to another. The dominant allele always is expressed even if combined with the recessive allele, and it's always written in uppercase letters. So for a dominant tall, you could have a big T, which would be your what we call homozygous true or homozygous dominant. And so it basically would show the trait. And then you could have big T, little t would be your other choice, which would be heterozygous dominant. Your recessive traits were only expressed if the dominant allele was not present, and they were written in lower cases. When you start doing your crosses, you're going to write them in lower cases. And that would represent the trait that's usually masked. So more vocabulary. Genotype is your genetic combination. So those will always be written in letters like big T, big T, big T, little t, or little t, little t. Your phenotype is descriptions that are physical characteristics or trait. So they'll always be written with words like tall, short, pink, purple. Those would all be your phenotypes. Homozygous means that both alleles are the same. So for us, it would be big T, or little t with the crosses that Mendel just did. And we'd call this true breeding. If it has one of each letter, like big T, little t, that's heterozygous. And then hybrid would be where you have crosses of parents with different traits. So the offspring would of big T, and little t would be like big T, little t. I'm not sure what's happening to my video here. So the next law that we need to talk about is our law of segregation. And so this is when gametes form, the alleles from each parent are segregated or separated from each other. And then each gamete carries a single allele from each gene. So this is what it would look like. You have your parents, big T, and then little t, and then you would do your crosses out to find out what your different generations would be, and you would probably end up with a certain probability, and that's your law of segregation. And that's what you have there. Okay, so Punnett squared is how we decide what offspring we would have by doing our different crosses. So instead of doing all that hard work for each of the possible outcomes under the law of segregation, we can do Punnett squares. So we use the genotypes in this particular process in order to find out what our offspring would be. My computer's acting up, guys, so I'm sorry. So for this cross, they had the mother was big T, little t, and the father was big T, little t. So they both are heterozygous, and they both are dominant. So what you do is you bring down a letter from the top and then you bring across the letter from the side. So this offspring would be big T, big T. This offspring is big T, little t. This offspring is big T, little t. And this offspring should be little t, little t. So this is a mistake right here. So make sure you realize that. So there is a 1 in 4 chance of the offspring being big T, big T. There is 2 in 4 chances of the offspring being big T, little t. And there is a 1 in 4 chance of the offspring being little t, little t. So 3 out of 4 plants are tall and 1 out of 4 plant is dwarf. So the ratio is a 3-to-1 ratio. So then we have what's called the law of independent assortment. And genes for 2 different traits are inherited independently. There's no connection between them. They don't have a set number where you're going to go and cross this and this. It's going to happen on its own. And that's why it's called an independent assortment. Okay. It's not wanting to move forward. I've never had this happen before, so I'm sorry. But it's not progressing the way it should. Okay, I think this is the end of it. All right, so we will be talking more about Punnett squares in just a brief few minutes and the next video that you see. And then they're going to be talking about some different traits that can be created from you crossing different things. So I hope you guys have a good day.